The second part is the synthesis stage. The second part is the synthesis stage. So synthesis basically means the combining of parts to make a whole. Synthesis means the combining of parts to make a whole. Combining of parts to make a whole. That is what we mean by synthesis. So during the S stage or the synthesis stage, the cell makes copy of its nuclear DNA. The cell makes copy of its nuclear DNA. Copy of its nuclear DNA. So it has to do this. It has to make copy. In eukaryotes, DNA is located in the nucleus. So we know that in eukaryotes, DNA is located in the nucleus. In eukaryotes, the DNA is located in the nucleus. So during interphase, if you looked at the DNA is loosely organized and appears grainy in photographs. But by the end of the synthesis stage, the cell nucleus contains 2 complete sets of DNA. By the end of S stage, the cell nucleus contains 2 complete sets of DNA. So we will go through a short video to show you what DNA replication looks like, though we will not discuss in details because we will be discussing that in another unit. So this is how the DNA looks like. It is first twisted in a double helix. It's twisted in a double helix. But before it replicates, it opens up. After opens up, if you look at the DNA, it has this complementary bases, cytosine and guanine, adenine and thymine. And it has, you can see all of them. If we have C here, we will have a T and so forth. So they are complementary. They are complementary to each other. So what happens before the DNA replicates? An enzyme comes and unzips or opens the structure of the DNA, that is the enzyme helicase, making a replication fork. So after that, what happens is that another one called primase will attach what we call a primer. Remember, now we want to make another copy of this DNA, and this cannot start unless there is a primer attached there. Then another molecule will come, the DNA polymerase, and it will start attaching now the bases or the DNA bases, of course, as from 5 to 1. That is the leading strand. So in the leading strand, this is made continuous. This is made continuous. They will be attached like that. But we still have another lagging strand, this one, which starts from 3 to 5. In this case, it will not go the same way, but a fragment will be attached there, what we are calling the primer. So the primer will be attached here to form Okazaki fragments. So the DNA polymerase will put the bases from 5' to 3'. So it will attach. After that, DNA polymerase will organize the whole thing and fix the bases. Then exonuclease will come and remove the primers that had been attached here. Then DNA polymerase will attach new DNA bases. So the whole structure will now be a complementary of the other one. So we have here DNA ligase. So you can see at the end we are having 2 DNA molecules which can recoil back into their double helix structure, which can, so we didn't need to go into these details, but it is good to learn them so that we know what we mean when we talk of replication. We also talked about the cell cycle control. So you can see that we have proteins called kinases. So as from G1 to S to G2, we will always meet these proteins which will be, of course, controlling the division of this, the cycle, either allowing it or stopping it. So these signals will either allow it to continue or to make it stop and restart afresh. So there is one signal here and there is another signal here. So it may come up to here and does not enter the S phase, or it may come up to here and doesn't enter the G2 phase, or it may come and doesn't enter mitosis. So depending on the conditions of the cell, if the conditions are good, then the cell will divide. But if conditions are not good, like if the DNA wasn't replicated, then this will be stopped. This will be stopped. Or if the DNA had some default, then it's better for it to be checked and that the cell doesn't enter into cell division. So this is what we mean by controlling the cycle or having checkpoints. So these checkpoints ensures that the cell goes through the right cell division procedure and that it has all the required materials. In gap 2 or G2, which is the third stage of the cell cycle, the cell continues to carry out their normal functions. The cell continue to carry out their normal functions. Their normal functions. When we talk of them carrying out their normal functions, what are we talking about here? We are basically meaning that it will continue doing its right task. If it is a skin cell, it will continue protecting the organism from invasion by bacteria and such stuff. If it is a blood cell, it will continue transporting materials within the blood, like, for example, oxygen being transported by the red blood cells and so forth. This stage also includes critical checkpoint. This stage also includes a critical checkpoint. A critical checkpoint. Just like the ones we've been discussing just in a moment. Normally, everything must be in order or adequate in order for the cell to enter into mitosis. So the cell must be adequate in size. The DNA should not be damaged and such stuff before the cell goes through mitosis and division. So it can't go into division if it is having some defects or defaults. So if it didn't have the right, so everything must be in order. Before the cell enters mitosis. The next stage we will be looking now at is mitosis itself, or the nuclear division. So mitosis, which is of course which is the fourth stage of the cell cycle, includes two processes. It includes two processes, and these two processes that are included in this is mitosis and cytokinosis. Mitosis and cytokinosis. So cytokinesis, these are the two main processes that are involved here. Mitosis is the division of the cell nucleus. Mitosis is the division of cell nucleus. Mitosis is the division of cell nucleus. And its contents. And its contents. The division of cell nucleus and its contents. During mitosis, the nuclear membrane dissolves. So during mitosis, the nuclear membrane dissolves. That is step number one. The nuclear membrane dissolves. The duplicated DNA condenses around proteins. The duplicated DNA condenses. The duplicated DNA condenses around proteins and separates, and the two nuclei form around proteins. So it will condense around proteins and separates and new nuclei forms. So new nuclei will form. And lastly, we have now the cytokinesis. Cytokinesis is the process that divides the cell cytoplasm. Cytokinesis is the process that divides cell cytoplasm. So in the first instance, mitosis only divides the nuclear. It divides cell nucleus. In the second case, we have cytokinesis, which is the division of the cytoplasm. So after the cytoplasm has separated, the result is two DORA cells. So we will continue with our discussion in the next video about what happens, and we will see a video also to make us understand better.